Good day everyone, we are the group 6 and for today we are going to have the post-laboratory report of the exercise number 7 which is entitled The Analysis of Milk for the Lipids, Carbohydrates, and Protein. Introduction. Milk is an emulsion or colored of butterfly globules within an aqueous fluid. Each fat globule is surrounded by a membrane consisting of phospholipids and proteins, which emulsifiers that keep the individual globules from joining together into noticeable grains of butterfat. The largest structures in the fluid portion of the milk are casein protein micelles, aggregates of several thousand protein molecules, bounded with the help of nanometer scale particle of calcium phosphate. The casein protein make up around 80% of the protein milk by weight. The type of carbohydrates found in the milk structure is lactose, which is the combination of the glucose and galactose, which gives milk its sweet taste and contributes about 40% of whole calories of milk. Human milk contains on average 1.1% of protein, 4.2% of fat, 7% of lactose, and supplies 72 kilocalories of energy per 100 grams. Cow milk contains on average 3.4% of proteins, 3% of fat, and 4.6% of lactose, 0.7 minerals, and supplies 66 kilocalories of energy per 100 grams. For the objectives of this laboratory activity, firstly we have students should be able to determine the presence of fat in the solution and to determine the percentage of protein in whole milk.
For part 2, determining if the milk fat contains unsaturated fat. To the test tube with the methylene chloride and removed fat, add 3 drops of bromine solution. Record the color of the solution in the test tube after the bromine has been added. For the result is, after adding bromine, the solution turns a violet tint. And for the question is the fat contained saturated or unsaturated? Explain. The fat contained unsaturated, since the presence of protein in the specific sample is indicated by the test tube violet color. Is there any other chemical test to determine the unsaturation of the structures? If any, explain the test briefly. There is, and it's called bromine test. It is a test for the presence of unsaturation, carbon to carbon, double or triple bonds, phenols and anilines.
Part 3. Determine the percent protein in whole milk. Weight of the milk is 2 grams. The weight in grams of the watch glass is 50.498 grams. The weight in grams of the watch glass with milk is 52.498 grams. Weight in grams of the milk is 1.5 grams. So to get the percent composition, we have to determine the true value minus experimental value divided by true value times 100. So the percent composition of the milk from the filter paper is 95.19%. What happens when you add the concentrated acetic acid into milk solution? The product of casein increases as acetic acid concentration does. Protein bonds will be hydrolyzed by acetic acid into less complex peptide linkages. More simple proteins dissolve in water as a result of this alteration, increasing casein yields.
Why Burette test is used to determine the absence or presence of proteins. Explain by giving your reasons and reaction if necessary. The Burette reaction can be used to assess the concentration of proteins because peptide bonds occur with the same frequency per amino acid in the peptide. Are your observations consistent with your expectations? Explain briefly. No, because the solution occasionally alters from time to time.
And for our conclusion, Through our lab experiment, we discovered that adding bromine to milk causes it to turn violet, which is a quite unusual hue for milk. Additionally, we discovered that because peptide bonding happens exactly once for every amino acid in a peptide, the Biuret reaction may be used to measure the concentration of proteins. Simple carbs are tested for using Benedict's test, with free ketone or aldehyde function groups reducing sugars, monosaccharides, and some disaccharides are recognized by the Benedict's test. Urine glucose levels can be checked using Benedict's solution. That's all for our report. Thank you.